Okay, mitochondria are the powerhouse of the cell. So you must have used the same textbook I used when I was in biochemistry or molecular biology or cell biology so many years ago. One of the functions of the mitochondria are to produce what? ATP, cellular energy, a molecule that most cellular processes use to produce energy. Why is energy important? This is perfect for an 8 a.m. class. Why is energy important? What does it do for an organism? What happens if you don't make ATP?
No, break down the food, or
If you don't break down food, if you don't produce cellular energy, what can't you do?
Activity process.
Anything. It's cellular processes, activity. Right? You wouldn't be breathing right now, you'd be dead if your cells weren't making ATP. If the mitochondria weren't processing the cellular products that they break down and produce ATP. We have in these nematodes all have mitochondria, like most or all animals. And one of the things about mitochondria is they all have their own mitochondrial genomes, which we'll refer to as mtDNA, mitochondrial DNA. Did you know that? I never learned that when I was an undergraduate, so I'm always surprised when people know that, so good. Do you know what proteins do electron transport in the production of ATP? There are a series of 5 electron transport chain complexes.
like cytochrome A.
Cytochrome C, oxidase, and there are a bunch of different proteins. I'm not asking you to actually tell me. I'm glad you remembered one of them, though. There are 5 different protein complexes that pass electrons from one complex to the next, and then at the end, protons are pumped to create an electrical gradient. Okay, that's not the critical part of this aspect of the project. But what you need to know is that every one of these complexes, let's model complex 1, has some proteins, should probably make that a little bit more blobby looking, that are encoded by the mitochondrial DNA. And every cell, of course, has a nucleus with a nuclear genome. So I'm just going to model 2 nuclear chromosomes as lines instead of circles. And those nuclear chromosomes also encode proteins that get into the mitochondria and fit together with the mitochondrial encoded proteins to produce each one of these complexes, except for complex 2, that's not important. So complex 1, complex 3, complex 4, complex 5 are all combinations of proteins that are encoded by the mitochondrial genome and by the nuclear genome. And they all have to fit properly together to actually carry out the function of that protein complex. If the proteins don't fit physically perfectly together, as in this model, which might be complex 1, for example, it doesn't matter. It's just illustrating the point that the 2 genomes, the mitochondrial genome and the nuclear genome, which we'll call nDNA, that's supposed to be an N, lowercase n, those genes have to work together, their protein products have to work together inside the mitochondria to do electron transport and produce ATP. So in this case, they're fitting together well, you might get ATP production out of the mitochondria. But what happens if there's a mutation? This project is the perfect combination of genetics meets cell biology, really, I think, in my opinion. What happens if there's a mutation in one of these red genes, the ones that are encoded by the nuclear genome? What does a DNA sequence change potentially due to the protein product? If it changes an amino acid, could cause a change in the shape of the protein. So if that gene encodes this protein, and the wild type version, no asterisk, is shaped like this, it could be that that mutation causes a change in the shape of the protein, so that now these two proteins can't actually fit together properly because that one's too bulgy there and that one's too bulgy there, and they're bulgy, and they can't actually occupy the same space at the same time because this isn't science fiction. So the basic point then is that DNA sequence differences or mutations in either the nuclear genome or the mitochondrial genome can cause changes in shapes of these proteins, and that can cause them not to fit together to produce a functional electron transport chain complex. And if that happens, then you might expect you don't make ATP. Maybe. It's one possible outcome. That's the theoretical background of mutation, DNA sequence differences, and effects on mitochondria. Now, it's known in humans, and in most species, in fact, that there are mitochondrial mutations that cause diseases. So in humans, there's a huge host, there's like 10 or 20 or 30 different diseases you can get if you have mutations in your mitochondrial DNA that essentially do this. They change the shape of your proteins in your mitochondrial electron transport chains. ATP isn't produced as efficiently. And so what sort of systems do you suspect would be affected by mitochondrial mutations? Like which systems in the body? Muscles are the main target because muscles are the ones that use a lot of ATP to help us move. Heart is particularly affected. Eyes are occasionally affected. Musculature certainly. It must be early. So there's this correlation between, well, if you've got a mutation, maybe you're not making enough ATP, so you don't have good musculature, your heart has problems, your eyes have problems. One thing that can also happen is if you don't have ATP, you don't grow as quickly because your body doesn't have a lot of energy to make new cells. That is the hypothesis that drives the research project that we're going to be working on. That if you have mutations in the nuclear or the mitochondrial genome that cause mitochondria to not function properly, then we might discover that those mutations cause slow development or slow growth. So let me introduce again the biological players in the system. Two different strains of C. briggsae, named after Margaret Briggs, who was a graduate student at Stanford and found these worms on the Stanford campus. We have two different strains, so you can think about them as two different populations, two different parts of the world. HK104 is a version of C. briggsae that was found in Japan. And AF16 is another population of C. briggsae that was isolated from a garbage heap in India. It turns out most C. briggsae are found in compost piles. They like to eat bacteria that grow on rotting fruits and vegetables. So they're almost always found in fields, vineyards, or garbage piles, compost heaps. These are two members of the same species. Two different populations, but they can have kids together. But they're genetically diverse. They're about 1% different at the sequence level. That means about 1 in every 1,000 base pairs is different. It's pretty genetically diverse, it turns out. Do you know what that number would be for humans, for any pair of us, if you compared our DNA sequences? We're about 1 in 1,000 base pairs different, so 0.1%, any two of us, on average. So this is pretty genetically different for an animal species. And so one of the major arms of research in my lab is asking, are these two populations kind of becoming different species because they're getting so genetically different that maybe someday they won't be able to have hybrid offspring if you mate HK104 worms with AF16 worms. That's totally related to this research project that we're going to have you embark on today. So HK104 have mitochondrial genomes and they have nuclear genomes. Mitochondrial DNA is circular, nuclear genomes kind of looks like the Starship Enterprise, for those of you that are Trekkies, although that's kind of a dated reference. And AF16 has red, color code these red because they're from the tropics, and blue because they're from a temperate area. So they have mitochondrial and nuclear genomes that work perfectly together to make functional mitochondria, functional MT. So we know at the start that each of these two parental populations, or wild strains, are healthy. They don't have any issues that we know of anyway, producing ATP, growing. So we consider these sort of the baseline. These are wild type, normal, healthy worms. But again, we know that they've got DNA sequence differences that distinguish the blue chromosomes from the red chromosomes. This is how we know we can color code them as different colors. That represents the fact that these chromosomes have specific sequences that aren't the same as those. So for example, you might have an A, that's not a double helix, by the way, that's two different homologous chromosomes. There might be an A at that position on that chromosome, and there might be a G at the same position in AF16, so we can tell the two apart. Likewise, in the mitochondrial genome, there might be a C there. There might be, and again, I'm just making this up, a T there. And based on that previous drawing, it might be that you have to have that combination, that specific combination of alleles, mutations, to get that nice fit between proteins that are in the electron transport chain. Whereas if you have A's and T's, those might be totally different shapes of those same two proteins. So we call this co-adaptation or co-evolution, the compensatory mutation. If you have a T, you have to have the A that provides the right shape in that gene to have a functional mitochondria. So here finally is the question that your data are going to help us answer, and I'm really looking forward to seeing what you discover. What would happen if you had the HK104 nuclear genome and you combined it in an organism that has AF16's mitochondrial genome? That is, you take, in this case, the hypothetical C, and you combine it with the A. Or you take this protein and you combine it with that protein. What's going to happen to those worms? Can we detect that there's any difference at all between these hybrids and their parents, HK104 and AF16? And in this case, these particular worms are called cybrids. It's a type of hybrid that comes from cytoplasmic hybrid. That is because the mitochondria come from the cytoplasm and the nuclear DNA not from the cytoplasm. So it's a class of hybrid that's called a cybrid. It's a worm. It's just got this funky genetic makeup that we created, and Shadi actually was the one who created these strains of worms that you're going to work with. So we have some that have HK104 nuclear DNA with AF16 mitochondria, and we have the opposite. We have ones with HK104 mitochondria and AF16 nuclear DNA. And these strains, we have three. They have names, FB59, FB60, and FB61. And these are, we have three different versions of that genotype, FB62 through 64. And we have the parents, AF16 and HK104. So the goal is we want to try to find out something about the health of these worms, the cybrids, compared to their parents, AF16 and HK104. Are they less healthy in some way? And what I'm proposing to you that we do is we use growth as a way to determine health. We're going to measure how long worms are at various developmental stages, various different time points. So simple experiment, but really powerful. So here's the goal. When it comes down to what you actually do, you're going to collect larvae from all of these strains. So we've got the plates of worms for you. So there are 6 experimental hybrids plus the 2 parents. Collect our larvae, they all have to be the same age, because we want to be able to make sure we're not comparing apples to oranges. We want to start all these worms at the same age. That's a process we call synchronization, which Shadi will talk to you about more. So we want synchronized larvae. They're all the same age, and then take pictures of them at regular time points, and then we can use those digital photographs to then later measure the lengths of worms of all of these 8 strains as they grow. And then at the end, the analysis is there a difference in growth rate between the different strains? So today, my understanding is the goal is to practice picking worms. We have to have, this is really important, 1 worm per petri dish. And these worms need to be named, because we want to look at one worm as it starts out as an embryo and becomes an L1 larva, and then an L2 larva, and then an L3, and an L4, and an adult. When you take pictures every 24 hours or so, we need to know that you're looking at the same worm through time. So this needs to be worm 1 of AF16. And then you need to have a second embryo on a totally different petri dish so you know which one you're working with that's worm number 2 of AF16. Or you can name them Peter and Frank and George and Bob or whatever you want. It doesn't really matter. They don't have to be numbered. So that's going to be one of the most critical things that we do. Today you pick worms on individual plates, practice taking pictures of the worms, and practice doing the length measurements using the computer. And then next week, it's full-on data collection. So this week you get a trial run to work out any kinks to understand the process, and then next week it's great guns, here we go, let's do the experiment, and then we'll discover what we find.